Okay, so this week's interactive case study is on how does penicillin kill bacteria, and what have bacteria evolved to do. So the first thing we want to look at is the cell permeability, and we need to keep in mind that the bacteria that we're mainly concerned with at this point are the gram-positive bacteria. Those are the ones with the peptidoglycan in the cell wall, not the gram-negative with the LPS. So you want to look at the enzymatic inhibition mechanism and understand the structure of the bacteria and the cell wall, and that humans don't have peptidoglycan in our cell walls. So what the penicillin prevents is that final cross-linking step, the transpeptidation, and this creates super weak, unstable cell walls in the said bacteria. And so these fragile bacteria cell walls burst, they break open, bacteria flies out, and they die. And so luckily, penicillin doesn't do anything to healthy non-bacterial cell walls in humans, and it doesn't disrupt any of those normal cellular processes. Okay, so when we look at the enzymatic inhibition in slightly more detail, I guess, let's see if I can move this. Sure. Um, I just had three other examples, um, kind of more specific ones than this, the basic one. And so if you look at the antibiotic or the drug tetracycline and how it inhibits ribosomal synthesis and blocks protein synthesis in the bacterial cells, so hence the bacteria dies. If you look at ciprofloxacin or Cipro, it's often called, this antibiotic, its particular specific target is the DNA gyrase. And so it doesn't target human gyrase in human DNA, but it does target the gyrase in the bacterial DNA. And then third example is sulfamethoxazole. I think that's how you say that. And that inhibits the enzyme dihydroelectrate synthase, which synthesizes folic acid. And we know that folic acid is necessary for all cells. And so without the folic acid within the bacterial cells, these cells die. They can't proliferate. I've got a couple more slides. So how has bacteria evolved now to become resistant? Well, bacteria are prolific reproducers, so they're highly susceptible to genetic mutation. And so they can just mutate, and the antibiotics quite simply won't be effective. That's the wrong effective, sorry. And then it can be due to overprescription or misuse of the antibiotic. It can also happen through bacterial conjugation. Probably remember the pilus. And even it can happen when the bacteria can adapt and sort of make their own beta-lactam ring that's resistant. So some bacteria examples that are highly resistant to antibiotics that we currently have. So nearly all of Staph aureus, we're on second generation. We've got methicillin resistant bacteria. We have drug-resistant tuberculosis, TB. We have vancomycin resistant enterococcus, and we have carbapenem resistant enterobacteria, I think it's called, if I can grab this. Well, and then one of my last slides is I just wanted to show that there's a chimera image, and then the bound structure of penicillin. So you can see the penicillin. The image on the right shows cephalosporin and a small peptide linked. So what normally would cause the form the crosslinks, but with the antibiotics, the penicillin or the cephalosporin, it cannot form those crosslinks. So when you look inside the molecule, you can see a tiny peptide chain inhibiting that process. And I think the last slide is just the works cited.